This video is going to focus on translation, which is actually bringing a molecule of RNA into a protein. So the previous video explained what RNA was, and transcription, which is the process of making DNA to RNA. Translation is actually making RNA into a protein. If you think of translating, it is generally when you go from one type of language to the next. This is very similar. You're going from nucleic acids to amino acids. But before we begin, I want to review what a protein is. And hopefully you remember that it is a macromolecule, and they're amino acids that are held together by peptide bonds, which are condensation reactions. And a polypeptide, there are hundreds or thousands of these amino acid sequences. Now, the sequence of the amino acids actually determines the type of protein that you just made, because there are four main levels of structure. So you start off with just the primary structure, which is just a sequence of amino acids. And then, due to that sequence, there's certain hydrophobicity, and each level you actually get different bonding. So it depends on the hydrogen bonds and all of that, which is determined by the sequence of amino acids and how closely they are. So the sequence, it always goes back to actually the sequence of amino acids, and then it turns into your ultimate globular shaped protein. So the actual code of the translation is very important, and it's the genetic code, which is actually nearly universal, which between all organisms, which is slight evidence for evolution. And it translates the mRNA into proteins. And it is set up in what's known as codons. And codons are 3 amino acids. And it's kind of like if our language only had 3-letter words. We have 26 letters in our alphabet, and if we only had words that were made up of 3 letters, similar to the genetic code, and their words are called codons. So if you look at a strand of DNA on the top in the blue, you have AAA, and then when it's transcribed into RNA, you get UUU, and then that UUU codes for a particular amino acid, and that's how you actually translate your RNA into a protein. And the codons, as I said, are a series of 3 mRNA nucleotides. So they each are for a specific amino acid. Now they have actually figured out what's what. So the genetic code table is how you can actually tell what amino acids are being coded for. So you'll see on the far left, those are the first letters, the top are the middle letters, and then the right side are the end letters. And there is what's known as a start codon, AUG, which if you look in the code table, it actually codes for methionine. So that is actually, if you remember back to transcription, the promoter. The promoter is synonymous. The promoter codes for the start codon, which then makes methionine. So every single one of your amino acids chains is going to start with methionine. And then there are 3 stop codons, UAA, UAG, and UGA. And you'll see them in our table, and they just say stop next to them. And those are what's known as your stop codon. So it makes sure that the gene is ended. So now before we get into the actual translation, I'm going to review the types of RNA. First, the mRNA that actually brings the DNA code out of the nucleus to the ribosome. tRNA that brings amino acids in the cytosol to the ribosomes because they have specific bonding sites for your amino acids and have the anticodon that pairs up with the mRNA codon for a specific amino acid, and then the rRNA that is found in your ribosome. So translation occurs in a few different parts. The first 3 are very, very important. mRNA has been transcribed and leaves the nucleus through the pores. Then the mRNA goes and attaches to a ribosome, and it's always at the start codon, as you see AUG. Then what happens is tRNA, UAC, because those are complementary to AUG, pairs with it and brings the amino acid methionine. So as you see in the picture, you have the first part of the ribosome attaching, and then you have the tRNA coming in and attaching the end of the ribosome, and then you'll see you have 3 different sites in your ribosome, and the P site is where your methionine tRNA is right now. Then what happens next is the RNA moves along to the next codon and reads, and then tRNA brings the correct amino acid. So if you'll see you have methionine, and then next to it, the next codon is GUC, and the complement of that is CAG, and therefore val is brought. And you'll see that then they're bonded together, and then it moves along, and that UAC tRNA can leave. And then the next one will come in, and it just continues and continues and continues, adding on more and more amino acids as the codons are read. So once it reaches your stop codon, and this is why the stop codons don't need or can't have those amino acids on them, is when they come in, they're the release factor because there's no more amino acid to attach to. The amino acid chain will get released, and everything breaks up, and you end up with a polypeptide chain, and your mRNA actually has to go and get destroyed, or else it will continue to make more of the same polypeptide chain, and then you end up with too many proteins. But then that polypeptide chain actually folds and makes an actual globular shape and becomes its actual protein. So that is the whole process, and how each sequence of the RNA, the mRNA, codes for that actual amino acid. Because if it was something else, then it would code for a different amino acid chain. So this just gives you the picture of the whole thing. You'll have the DNA that makes the rRNA, mRNA, and tRNA, and then through translation, it's actually made from the RNA into your actual protein. So this just gives you the whole big picture. To go further with this whole thing, you can look at how mutations actually interrupt and how they would mess up the actual amino acid chain, and how this is actually controlled in your body. The next video we will be looking at how it's controlled more specifically, because the DNA wants to be able to control what's being made, so therefore we need some sort of way of turning these on and off and determining when each protein is going to be made.